Hi guys, Jason Matthew here, and today we're going to be looking at amino acids and proteins. I've broken up this lecture into a series of parts, and this is part 1, so please look out for the other parts coming soon. Now this information here about amino acids and proteins is more for a level 1 university course. So it's just an introduction, and we look at the basic information needed that you'll be required to know for amino acids and proteins. So what we're looking at here is a typical amino acid, and this could be described as a general formula for the amino acids. You see that there is a carbon that is connected to a carboxyl group, an amino group, and a hydrogen. All amino acids will have these three groups connected to a carbon: a carboxylic group, an amino group, and a hydrogen. Where amino acids differ would be with this R group. Alright, so amino acids differ because they have different R groups attached to them. Now we could actually categorize these amino acids based on the type of R groups that are present. So one category could be the non-polar aliphatic R groups. Alright, so they're non-polar, meaning that there's no charge or anything like that. And it's aliphatic means there's some typical alkyl R groups, carbon and hydrogens only. Now let's look at glycine here. So you see all of them will have the carboxyl group, the amino group, and a hydrogen. All amino acids will have that. And where they differ is where it's shaded here. This is the R group. Now the smallest amino acid possible is when the R group is hydrogen. And when the R group is hydrogen, that amino acid is called glycine. And glycine is the smallest amino acid possible. Now as you increase now, so like you go from an R group where an R group was hydrogen to an R group which is CH3 or a methyl group, then that's alanine. And that's the second smallest amino acid. So the smallest amino acid is when the R group is hydrogen, and that amino acid is called glycine. And you see here leucine and isoleucine and methionine, valine. And I left proline for last because proline is special. If you watch all the other R groups, alright, they are only connected to the alpha carbon. But with the R group of proline, you see that it's connected to the alpha carbon, but then it forms a ring structure by connecting to the amino group. So that is just something to note. Now please note that this amino group here is not part of the R group. The R group for proline is CH2, CH2, CH2. The only thing that is different from the other amino acids is that this carbon here, it completes a ring by combining, or yes, well, bonding with the nitrogen of the amino group. Alright, so these we have broken up the 20 common amino acids into different categories based on their R groups. So this here that we just look at were the non-polar aliphatic R groups. Now we have the polar uncharged R groups. So they're polar molecules, but they are uncharged. One example is serine. Now remember, all amino acids will have a hydrogen connected to the alpha carbon, a carboxyl group, and an amino group. Where they differ is the R group. So the R group for serine is CH2OH. Alright. And I would recommend that you learn serine, cysteine, and glutamine. Alright, let's look at cysteine. Cysteine has CH2SH, this sulfur hydro group, and we'll see the importance of that very soon for cysteine. And then you have glutamine, which is CH2, CH2, C11O, NH2. Alright. Now, why we say that these are polar is because, just a little chemistry on the side now, this oxygen, it is much more electronegative than carbon or hydrogen, thus giving a polar bond being formed. Alright, this oxygen again, it differs in electronegativity compared to the carbon and the hydrogen. sulfur, and for this example, you have both nitrogen and oxygen present. Aromatic R groups, we have phenylalanine, tyrosine, and tryptophan, all very common amino acids. And basically, why we call them aromatic R groups is because the R group has an aromatic ring. An aromatic ring is basically where you have double bonds in a conjugated form. Alright, so you have double bonds in a conjugated form. What some people will say that there's a benzene ring present, like in this one and in this one as well. Then you have the positively charged R groups. So for instance, you have lysine, arginine, and histidine. Now, we say that these are positively charged because if you look at the nitrogen in lysine, it has a positive charge, and this amino group is found in the R group. Alright? So the R group for lysine, for instance, CH2, 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 NH3, and it's plus. Similarly for arginine, there's an NH2 group here that is plus. Now for this one again, for nitrogen to have this positive charge, it must have 4 bonds present. Alright, so for instance, you see this nitrogen is bonded to this carbon plus 3 other carbons, right? And because it has those 4 bonds, you have a plus here. Now, for this one, the histidine here, what they should have really done was really put a hydrogen on this nitrogen here. So let's do that now. Alright. So if we was to put a hydrogen here, right, then this nitrogen becomes positive. Alright, so that's what it should have really been there. So that nitrogen should have been positive there. Alright, because there's a hydrogen, so it'll have 1, 2, 3, 4 bonds. So just like how we have positively charged R groups, we also have negatively charged R groups. So again, remember, all the amino acids will have the hydrogen, the carboxyl group, and the amino group. Where it differs is the R group. So for aspartate, it is CH2COO minus. Alright, it's acidic. Alright, so it's COO minus. So if you watch, the net charge for here will be negative, or 1 minus, because this negative and positive cancel, but then you'll have this negative here. For glutamate, it's CH2CH2, and then it's acidic group COO minus. So aspartate and glutamate, negatively charged R groups. So just to recap, remember all amino acids will have this skeleton here, hydrogen. This will be alpha carbon, and it's connected to a hydrogen, a carboxyl group, and an amino group. Where amino acids differ will be the nature of the R groups. And that is how we categorize or we classify amino acids based on what R group is attached. Now there are different ways in which we could write the name of an amino acid. So the long way would be to just give the whole name. So you have alanine, arginine, asparagine, and so on. Another way in which we could do it is that we use this international 3-letter abbreviation. So for alanine is ALA, for arginine is ARG, and we go on like that. So for most instances, luckily for us, the first 3 letters in the name corresponds to the 3-letter abbreviation. Alright, so that's for most amino acids here will correspond to that. And then if you find that the 3-letter abbreviation will take up too much time, there's good news for you in that you could use a 1-letter abbreviation. And not for all, but for a few of them, the first letter corresponds to the letter of the name. So like for instance, alanine is A. But then look at arginine, it's capital R. Lysine is K, and so on. So you have to be careful, right? Alright, so some very important biochemistry is taking place here. Now you can have 2 cysteine amino acids. So that's what we have here, alright? We have 2 cysteine amino acids. So this one here that has an SH group as part of the R group, and then this cysteine here has an SH group of its own. So these are two sulfur hydryl groups. Alright? Now these two sulfur hydryl groups could undergo a reaction where they lose hydrogen and they form what is called a disulfide bond. So this here is a disulfide bond. Now what you need to remember for this to really understand what's going on here is the definition of oxidation and reduction. Alright? Now there are different ways in which we could define oxidation and reduction. Alright, we could look at it in terms of the gain or loss of oxygen. We could also define oxidation and reduction in terms of the gain or loss of electrons. But for the purposes of this exercise here, we're going to be looking at the gain and loss of hydrogen. So let's look at reduction first of all. Alright, so reduction. You all remember what reduction is? Is it the loss or the gain of hydrogen? So think about it and get back to me. Let's go and look it up quickly if you can't remember. Right, so you should figure it out by now. Reduction is the gain of hydrogen, while oxidation is the loss of hydrogen. Alright? So what that means here is that when two cysteine, right, when these two sulfur hydryl groups, when they lose their hydrogen, when they react together by losing their hydrogen to form a disulfide bond, alright, so they're losing hydrogen, in this direction here, that's an oxidation reaction. Alright? Now two cysteine, when they combine together, they form one cysteine. Alright? Remember what I said this is? This here is your disulfide linkage. And that will be very important when we look at the different structures of proteins. In fact, in particular, when we look at the tertiary structure of proteins. Alright? Now, if you look here, we're seeing that there is reversible arrows here. What that means is that this reaction can be reversed. So, oxidation is the process where we lose the hydrogen, the two sulfur hydryl groups react, alright, and they lose the hydrogen, so there's oxidation process, loss of hydrogen, to form the disulfide linkage. Now, if you could reverse this process by doing the opposite of oxidation, which is to add hydrogen. So if you do the reverse there, in other words, you're adding hydrogen, so what are you doing? Correct? You will be doing a reduction. Alright? So if you do a reduction, so you're going from here back to here, that's a reduction process, that's a gain of hydrogen. So you're going to be reducing the disulfide bond, and by reducing the disulfide bond, you're going to be converting one cysteine to two cysteine molecules. Alright? So, let's just recap that again. Alright? Two cysteine molecules will undergo a reduction reaction where the two sulfur hydryl groups will react together and lose their hydrogens via oxidation process to form a disulfide linkage. And when two cysteine molecules combine, they form one cysteine. Now that whole process can then be reversed. If you reduce the disulfide linkage, so you undergo a reduction reaction, alright, the process is going to go in the reverse direction. So you're going to convert one cysteine molecule to two cysteine molecules. Alright. Another term you have to be familiar with is the non-ionic form and the zwitterionic form. Now look at the difference between the two. Alright, now if you watch, it's here. So the zwitterionic form is basically where you would lose the proton on the carboxylic group. So you're going to be losing this hydrogen here as a proton, and this proton then is going to come here, and the amino group is going to accept it. So let's recap. To form a zwitterion, the proton, the hydrogen on the carboxylic group is lost as a proton, and the amino group is going to accept the proton, or it's going to be protonated to form Na3+. And when you have the two charges here, we say this is the zwitterionic form. Alright. Now, out of the 20, there are 20 amino acids that basically most, if not all, your proteins in your body is made up of. Alright, so all the proteins in the body that is required for healthy human growth and health, alright, consists of 20 amino acids. Now, out of that 20 amino acids, some of them are what we call essential amino acids. So if you look on the left hand side here, you see that we put a list of the essential amino acids. And basically what an essential amino acid is, it will be those amino acids that we cannot synthesize on our own. Alright, and therefore we rely on our diet to get these essential amino acids. So that's the essential amino acids. I remember there are 20 amino acids that is required for proper health. Now out of those 20, about 10 of them are what we consider essential. And as I said before, essential means that we require them from the diet. We cannot synthesize them on our own. Now the non-essential amino acids, and sometimes the term essential could be very misleading. The point to note here is that when you add up all these, all 20 of them are really and truly essential for proper health. Alright? If you have a deficiency, for instance, in glutamic acid, right, if your body doesn't have enough glutamic acid, you will not be healthy. Alright? The whole thing here about the essential and non-essential is more to say in terms of nutrition. Alright? The essential amino acids, you cannot synthesize them, so you must get them from your diet. The non-essential amino acids, you do not require to get them from the diet. You can actually synthesize them on your own. And when we do the level 2 courses, you will see that we could use the carbon skeletons from certain keto acids and so on. We can remove the carbon skeletons and actually make our non-essential amino acids. So for instance, like there's some common ones, alanine. You could get alanine from pyruvic acid. You could get aspartate from oxaloacetate. Alright, so those are some of the more common ones, and you can see the other examples listed here. So you have a complete protein and an incomplete protein. The complete protein contains all 10 essential amino acids. And we see that animal sources are an excellent source for complete protein. So animal sources is the way to go in terms of getting complete proteins. One exception is that beans on the plant side, they contain some complete protein as well. Alright? Now the incomplete proteins, that is when this protein source, it lacks one or more of the essential amino acids. And that's very common when we look at most vegetable proteins. They are incomplete proteins. Alright, with the exception being beans, right? Beans tend to have a complete set of the all amino acids, alright? Now let's go to the lab now. Now in the lab, a test for amino acids is the Nonionic reaction. Now the Nonionic reaction reacts with amino acids to form a purple colored amino derivative. This derivative forms a useful test for amino acids, most of which are colorless. Alright, so let's just recap. A test for amino acids will be to use Nonionic. Alright? The confirmation is that the Nonionic is going to turn purple. Alright, so a purple color at the end of the test is a positive confirmation that there are amino acids present. And as the note says, amino acids tend to be colorless. Alright, so the ninhydrin will be able to confirm the presence of amino acids. And remember again that this is a test for amino acids. Now below here I've put some formulas and so on. You are not required to know that in terms of for the theory of the course, but it will come in handy if you have to eventually write up a lab report on the use of ninhydrin. Part of your lab report, part of the exercise in lab, you may have to use ninhydrin. Now, for most amino acids, the color is purple, and that confirms the presence of amino acids. What I want you to do is to do some homework and see if there are any amino acids that won't give you the purple color, but will give you a different color. So, if you find any like that, I want to know the names of the amino acids. I want to see different color from purple. So, there's something to check out, right? Not all amino acids will probably give you a purple color. They might have some that will give you a different color. So do some research on ninhydrin and see if that happens. Now, this is a very common MCQ question because some students could get confused. As I said, a test for the presence of amino acids will be the ninhydrin test. A test for the presence of protein is the biuret test. Now let's read and find out something about this biuret test. So remember, on this slide, we are testing for the presence of proteins. The ninhydrin test that we just looked at was a test for amino acids. Alright, so it's two different tests. One is a test for the presence of amino acids, and the other is a test for the presence of protein. In other words, you cannot use the ninhydrin test to confirm the presence of protein. Similarly, you cannot use the biuret test to test for the presence of amino acids. The ninhydrin test tests for the presence of amino acids. The biuret test tests for the presence of proteins. So you need to know the difference between these two tests. They are testing for the presence of two different things. Now the biuret reagent itself is light blue in color. So if you watch these two test tubes here, they have, all three test tubes have biuret reagents. But in these two, because there's no protein present, it remains its original color, which is light blue, pale blue. Alright? And the reason why it's blue in the first place is because it has Cu2 plus ions. Alright? Now the biuret reagent will turn purple when mixed with a solution containing protein. So for instance, if you look at the middle test tube on the other hand, while the test tube with starch and the test tube with water remain light blue, because if it just contains water or starch, there's no protein present. So the biuret reagent did not change color. But in the test tube here in the middle, if you look here, we see that there was a color change from the light blue to purple, and the reason for that is because albumin is a protein, and the biuret test turns purple when the solution contains a protein. The purple color is formed when copper ions in the biuret reagent react with the peptide bonds on the polypeptide chains to form a complex. Right, so we're talking about this peptide bond. Let's go into it some more. The peptide bond is a chemical covalent bond formed between the amino group of one amino acid and the carboxyl group of another. So just to recap, every amino acid will have an amino group and a carboxyl group. What's going to happen here is that a series of amino acids is going to come together, and the amino group of one amino acid is going to react with the carboxyl group of another amino acid, and via condensation reaction, a peptide bond will be formed. Long unbranched chains of amino acids can be linked together by peptide bonds to form oligopeptides. Oligopeptides could go up to about, let's say, 25 amino acid residues. Right? Polypeptides, we tend to use the term polypeptides for any chain of more than 25 amino residues and so on. Alright, so convention, so how do we write this down? Convention has it that polypeptide chains are written down from the free alpha amino group on the left, alright, to the free alpha carboxylic group on the right, and a hyphen between the amino acids to indicate the peptide bonds. Thus, the tripeptide, and you can put the NH3 plus, so that means that serine, alright, has a free amino group, the NH3 plus. It meant that the COOH of the serine reacted with an NH3 from the leucine and so on. Alright? So you always write going from the N terminal end to eventually the C terminal end. Alright? And normally we don't put the NH3 plus or the CO minus. We just write the amino acid present. So it puts, and we use 3 letters. So serine, leucine, phenylalanine. Right, so if you look here, alright, you have 2 amino acids, alright, and basically what will happen is that the carboxyl group of one amino acid is going to react with the amino group of another amino acid, and they're going to lose a water molecule because this going this way, it's a condensation reaction. And when the condensation reaction takes place, you form this here, which is called a peptide bond. Alright, so let's just recap here. So you're going to be reacting the carboxyl group with the amino group. Alright, so the carboxyl group of one amino acid with the amino group of the other amino acid. And when you're going in that direction like this, that's a condensation reaction. It's a condensation reaction because if you watch here, we're losing water. Alright? When that happens, you form this here. And this, as we saw in the other slides as well, is called a peptide bond. Alright? So this is how we go from, we form a peptide bond. It's between the carboxyl group of one amino acid reacting with the amino group of another amino acid, and via condensation reaction you're going to lose a water molecule to form a peptide bond. Now if you want, there's reversible arrows here, meaning that this peptide bond could have been hydrolyzed, meaning that you add water, so you could hydrolyze the peptide bond to form amino acids. So hydrolysis and condensation, they are very important in terms of condensation to make these macromolecules, and then hydrolysis is to break down these macromolecules back to their smaller components. Alright, we looked at condensation and reduction when condensation and hydrolysis when we looked at carbohydrates as well. Alright, so when you have a protein, any protein, the amino acid at the end, one of the amino acids at the end will have a free amino group, meaning that it's free to react with another amino acid if it's available. Well, that amino acid it's carboxylic and is going to react with this amino end. And on the other end of the protein, you would have the last, or if you want to call it the first amino acid. Well, the amino acid at this end would have a carboxyl group free. So we call this the carboxyl terminal end, or we could also call it the C terminal end. And we'll call this end the N terminal end. So you could call this also the C terminal end, and this you could call the N terminal end. Alright? It depends on, we call this the N terminal end because the amino group is free. We call this the C terminal end because the carboxylic group is free. Remember, we always go from the amino end to the C terminal end. Alright, so peptides. Peptide is the name given to a short polymer of amino acids joined by peptide bonds. They are classified by the number of amino acids in the chain. A dipeptide will be where 2 amino acids are joined by a peptide bond. A tripeptide is where you have 3 amino acids joined by peptide bonds. A polypeptide is a macromolecule containing many amino acids joined by peptide bonds. And finally, a protein is a biological macromolecule of molecular weight 5,000 grams per mole or greater, consisting of one or more polypeptide chains. Now, proteins are a very diverse biological molecules. They have many functions. One of them being that they act as receptors, so they send stimuli, example in neurons. They form channels, protein channels, like in the cell membranes. They control the movement of cell contents and so on. Alright, they also act as a transport molecule themselves. Classic example, hemoglobin in blood. And please do not terrify me by not knowing what it is that the hemoglobin transports. You should know that hemoglobin transports oxygen. Alright, in terms of storage, for example, ferritin in liver. And if you never heard ferritin before, please go and look it up. It's very common. But the word ferritin, it comes from ferrous, and that should be already, you should be already thinking of something along those lines. And I mean, it goes without saying, right, that life itself would cease to exist if we didn't have enzymes, right? Enzymes catalyze these biological, wonderful biological reactions that we are learning about. That is necessary for life. Structural, for instance, collagen in skin, and we probably look at some more examples later on in the course. Some proteins are antibodies, and they are very important in terms of the important role that they play in the immune response. Right, so just to recap, you have proteins that either act as receptors, channels, transporters, storage, enzymes, structural, and immune response. Right, so globular proteins, for example, enzymes. Now, globular proteins will have a variety of secondary structures. And if you're not too sure what a secondary structure is, don't worry. When we continue with amino acid and proteins, we're going to be talking a lot more about the different structures of protein, like secondary, primary, secondary, tertiary, and quaternary. Alright? Most, if not all, globular proteins will have a spherical, three-dimensional shape. And because of that shape, you will see later on that they are water soluble. Alright? They play important roles. Globular proteins play important roles in catalysis, regulation, transport, gene processing, and so on. Right? So these are some examples of globular proteins, for instance, hemoglobin and so on. Right, next type will be fibrous proteins, and these are our structural proteins. Right, and fibrous protein tend to have only one dominating secondary structure, whether it be beta pleated sheets or whether it be alpha helices. And if you are not too familiar with those names, don't panic. We're going to be coming to that in the other parts. Right, they tend to have poor water solubility, and that makes sense, right? Because like we're looking at structural proteins such as in the cytoskeleton, bone, and skin. And if that was water soluble, you would basically just dissolve away. So they are insoluble, right? Now, we're going to look a little bit more about this later on, but collagen, right, it has been studied a lot, and it's an example of a fibrous protein. Right, now the last one, membrane proteins, and I guess they will overlap with what we just did, but for instance, receptors and channels, they're inserted through membranes. You have they span the membrane. They could have extracellular domains and so on. They have poor water solubility, and they function in cell communication. Example, cell signaling and transport. So this is just an example of a membrane protein, the photosynthetic reaction center. So you see, it's a huge protein, and piece of the protein it spans the lipid bilayer, while some of the proteins is extracellular, while others are intracellular. So it's a big protein. Alright guys, so we are the end of part 1 of amino acids and proteins. There will be other parts being posted on YouTube. So what I would recommend is that you go to my YouTube channel. Alright, if you're not on the YouTube channel already, please do a search on YouTube for BiochemGM, and you will see the other amino acid and protein videos that will be coming soon. Alright, other than that, if you haven't done so as yet, if you don't know the drill, please join us on Facebook. We would love to hear the feedback and how we can improve this service for you all. Or, especially our international students, we would love to hear from you all. Alright, for our international students, if you are doing the course slightly different, if you have a different syllabus because you all look at other topics, I would love that you share that with us. Share any slides, any pictures you think might enhance what we are doing right now, because it's all about sharing, having this global network so that we could all improve. Because I will tell you up front, I don't know all the biochemistry, and there's definitely a lot of work that I could do still to improve what we are doing. So please give us that feedback. And as usual, this is Jason Matthew here, thanking you for listening about amino acids and proteins, and look out for more to come. So thanks again, and take care, guys. Bye.